Hi guys, let us continue with our topic HIV. Now you must be aware of its definition, classification, etiology, the way how it transmits. Am I right? Anyway, today let us take a look at the pathophysiology of HIV. Before going in detail, first let us take a look at its structure. So here is the structure for human immunodeficiency virus, which leads to AIDS. Okay, so our HIV is having a strong envelope. Okay, so this envelope is made up of lipid bilayer. So here you can see the lipid bilayer. So the strong covering of the human immunodeficiency virus is made up of lipid bilayer. Okay, so on the surface of the envelope you can see the projecting structures which are known as glycoproteins. Okay, and there exist mainly two subunits. They are named as GP120 and GP41. Okay, so the glycoproteins consist of two subunits. They are glycoprotein 120 and glycoprotein 41. So these glycoproteins helps the HIV to attach to the host cell membrane. Okay, and inside the envelope, let us see what are there. So inside the envelope you can see two types of proteins, that they are capsid protein and matrix protein. Okay, so inside the core you can see P24 capsid protein as well as P17 matrix protein. Okay, so the one which is marked in green color is the P24 capsid protein, and the one which is marked in blue color is the P17 matrix protein. So what is its function? It helps the virus to maintain its structure. Okay, so the glycoprotein helps the virus to attach to the host cell surface, whereas this matrix and capsid protein help them to maintain its structure. Okay, so inside the core you can see the most important part of the virus, that is the genome. Okay, so inside the core two identical copies of single stranded RNA viral genome is there. Along that you can see three enzymes. So here you can see the RNA strands. Okay, and along that you can see three important enzymes. So they are named as reverse transcriptase, okay, which is denoted as RT, then protease, which is denoted as PR, then integrase, which can be denoted as IN. Okay, so inside the core we can see the RNA viral genome, that too two identical copies. Okay, then along that we can see the three enzymes: reverse transcriptase, protease and integrase. Now you must have understood the structure of HIV. Now let us take a look at its pathophysiology in detail. To establish the infection, HIV must first attach to its host cell, right? So for that there should be an interaction must take place between glycoprotein 120 and the CD4 receptors on the surface of the host cell. Okay, so here you can see the virion is trying to attach to the host cell by interacting with the CD4 cell on the surface of the host cell. Okay, so our virus is trying to attach to the host cell membrane with the help of glycoprotein 120. So this one is having more affinity towards this CD4 cells. So what are CD4 cells? Cluster designation for cells, okay, for receptors. So these are nothing but the proteins which are present on the surface of T helper lymphocytes, monocytes, macrophages, dendritic cells, and brain microglia. So you can see CD4 receptors on the surface of all these type of cells. So this glycoprotein 120 subunit has high affinity for CD4 receptors, and this GP120 is responsible for the initial binding of the virus to the cell. Okay. Now, in addition to these CD4 cells, you can see there are co-receptors. Okay. So these co-receptors are known as, in this picture it's not that clear. It is separately shown. Uh, to comprehend that more clearly, I am demonstrating it here. So let us assume that this is the glycoprotein GP120 of HIV, and here is the CD4 cell, and these are the co-receptors. Okay, so this one and this one are the co-receptors. So in addition to the CD4 cell receptor, there must always be co-receptors, okay, on the surface of the host cell. The co-receptor is different for different host cell types. So once the initial binding occurs, the intimate association of HIV with the cell is further enhanced by these co-receptors, which are also known as chemokine receptors. So the main chemokine receptors are CCR5 and CXCR4. Okay, so these CCR5 and CXCR4 can be seen in lymphocytes as well as macrophages respectively. Okay, so the co-receptor which is found on lymphocytes are called as CXCR4. Okay, this is found on lymphocyte, whereas CCR5 are found on macrophages. Okay, so HIV that preferentially use this CCR5 co-receptor, so they are known as R5 viruses. Okay, so this R5 viruses is typically implicated in most cases of sexually transmitted HIV. So they are mostly macrophage tropic. And you can also see X4 viruses. Okay, so they preferentially uses CXCR4 as a co-receptor, and this is T cell tropic, that is T lymphocyte tropic. So clinical isolates may contain a mixture of this R5 virus and X4 virus, and some cases you can see viral strains are designated as R5X4. Okay, so this may be dual, that is they can be tropic, that is these viruses can use both co-receptors, whereas X4 viruses can only use CXCR4 and R5 viruses can only use CCR5. Is that clear? Now you must be understood the co-receptors as well as the CD4 receptors, right? So attachment of HIV to the cell, that is attachment of viral envelope and the host cell membrane, promotes fusion. So this is the second step, first step, that is fusion. Okay, So this results into the internalization of the virus, that is adsorption of the virus particle. So this process is mediated by glycoprotein 41. Okay, so the process of attachment of HIV to the host cell membrane promotes fusion, and this results in the internalization of the virus, and this process is mediated by glycoprotein 41. So what is the genetic material of the HIV? Single-stranded RNA, right? So this is a positive sense single strand RNA. So it is denoted as ssRNA. What it is? Positive sense single strand RNA. So now to replicate in the human cells, the virus transcribes this RNA into DNA. So what happens? As a result of the fusion, whatever be the genetic material, that is the RNA genome, is entered into the human host cell. Now for the replication, to replicate in the human cells, it tries to transcribe the RNA genome into DNA. So generally this transcription occurs from DNA to RNA, right? But in case of HIV, it works backward, that is from RNA to DNA is transcribed. Okay, RNA transcribes to DNA. So this, in case of HIV, we can give the name retroviruses. So the transcription takes place in the backward direction. Now for the transcription, HIV is equipped with a unique enzyme, that is reverse transcriptase, which is also known as RNA dependent DNA polymerase. Okay, so this reverse transcriptase helps the transcription of the RNA to DNA. So this reverse transcriptase, what it do? It produces it first synthesizes a complementary strand of the DNA using its viral RNA as a template. Okay, so by using the viral RNA as a template, this reverse transcriptase produces a complementary strand of the DNA. So the RNA portion which is produced, from which the DNA is produced, is then partially removed by the ribonucleases H. So here is the RNA which is released from the virion into the host cell, from which the reverse transcriptase produces the complementary DNA strand, and the first RNA part is removed by the enzyme ribonuclease H. So it is known as RNase H. Okay. So this allows the reverse transcriptase to complete the synthesis of a double-stranded DNA molecule. So now following the reverse transcription, after the reverse transcription, you can see the double-stranded DNA is formed. So following the reverse transcription, the final double-stranded DNA product migrates into the nucleus, and then it is integrated into the host cell chromosome by the enzyme integrase. So this is another enzyme which is unique to HIV. So first transcription from RNA to DNA is done by reverse transcriptase. Now the double-stranded DNA is integrated with the help of integrase in the nucleus of the host cell. Okay, so the integration of HIV into the host chromosome is troublesome for several reasons. First of all, HIV can establish a chronic and persistent infection, particularly in long-lived cells of the immune system, such as memory T cells, that is memory T lymphocytes. Second reason is that integration is random. Thus, making it difficult to target and extract the integrated HIV. And the last reason is random integration of HIV may cause cellular abnormalities and induce apoptosis. So, now you must have understood why integration of HIV into host chromosome is a troublesome process. So, in addition to this, there is activation of infected cell by antigens, cytokines, or other factors such as nuclear factor Kb. So, this is an enhancer binding protein. So, it is known as NFKB. Okay, so this NFKB normally regulates the expression of T lymphocyte genes which are involved in growth, but also can inadvertently activate replication of HIV. So when the replication is induced, the host DNA polymerase transcribes the integrated proviral DNA into messenger RNA. So the DNA particle which is integrated from double stranded to integrated are known as in proviral DNA. So this proviral DNA is converted to messenger RNA with subsequent translation of mRNA into viral proteins. Okay, so here you can see the proviral DNA is integrated into messenger RNA. Okay, so some of the functional proteins are formed by the cleavage of long proteins by the enzyme protease. So at first, transcription and translation are done at a low level, yielding various regulatory HIV proteins such as TAT, NEF, and RAVE. So these are the regulatory HIV proteins which are synthesized. So these TAT proteins, they are nothing but the potent amplifier of the HIV gene expression. Okay, so it binds to the specific RNA sequence of HIV and then initiates and stabilizes the transcription elongation. Okay, and coming to NEF, so there is no evident function, although it appears to downregulate class 1 molecules and protect infected cells from cytotoxic T lymphocytes. Whereas this REV protein regulates the post-transcriptional activity, and like that, it is essential for the HIV replication. And this REV essentially shifts the synthesis of HIV regulatory proteins to structural proteins. Example, glycoprotein 120. So by inhibiting the viral mRNA splicing and exporting the mRNA outside the nucleus. Okay, so this REV essentially shifts the synthesis of HIV regulatory proteins to structural proteins. Now the mRNA and the structural proteins are produced. You can see the structural proteins are produced. So now the last step is to assemble all of the structural parts of the virus. So this assembly of new virion particles occurs in a stepwise manner. So it begins with the coalescence of HIV proteins beneath the host cell lipid bilayer. So first of all, whatever the protein, glycoproteins and protein materials produced, they are adhering to the host cell surface. Then what happens? Then subsequently the nucleocapsid is formed with viral ssRNA and other components packed inside. Okay, so once all the elements are filled up, that is packaged, the virion then buds through the plasma membrane. So once all the elements are filled, what happens? It is separated from the host cell plasma membrane. Now you can see the new HIV virion is formed. Okay, so this is also having the same characteristics of the first HIV virion. Okay, so the HIV virion which stimulated the synthesis of the HIV produces a number of new virions. So HIV-1 exhibits an estimated 10 billion new viruses produced each day. Okay, so more than 99% of these viruses are produced in newly infected cells. Ultimately, most infected cells will be destroyed from a number of mechanisms, including cell lysis by newly budding virions, cytotoxic T lymphocyte induced cell killing, syncytia formation, or apoptosis. Okay, so these uninfected cells clump onto the infected cell and fuse into a giant multinucleated cell. Then the syncytium induced X4 virus phenotype may develop later in disease and is associated with more rapid disease progression. So destruction of the CD4 cells leads to compromised immune function and consequently AIDS. So in short, here is the life cycle of HIV. First step is the binding, then fusion, then reverse transcription. Fourth step is the integration, then replication, then assembly, and the seventh step is the budding. So now you must have understood HIV life cycle in short. Now let us move to the clinical presentation. First of all, clinical presentation of primary HIV infection in adults. The clinical presentation of primary HIV infection may vary, but patients often have an acute retroviral syndrome or mononucleosis-like illness. So these symptoms often last 2 weeks and hospitalization may be required for 15% of the patients. So mostly the symptoms you can see fever, sore throat, fatigue, weight loss and myalgia. And 40 to 80% of the patients will also exhibit a morbilliform or maculopapular rash, usually involving the trunk. Also you can see diarrhea, nausea and vomiting. Then symptoms such as lymphadenopathy, night sweats, aseptic meningitis, which may be characterized with fever, headache, photophobia and stiff neck. So this aseptic meningitis may be present in 1/4 of the presenting cases. And other important factors are, that is laboratory factors, are high viral load. So that may exceed 1 million copies per ml and persistent decrease in the CD4 lymphocytes. So these two are the main two indicators for HIV infection. Primary infection is often associated with a high viral load and development of an immune response that for a period of time suppresses but does not eliminate viral replication. During this period, HIV is trapped by follicular dendritic cells in lymphoid tissues and replicates in the germinal center. The amount of HIV RNA in plasma falls substantially at this point and symptoms resolve gradually. This decline coincides with the development of an immune response to HIV. So, in the clinically latent period, it is not virologically latent because HIV replication and immune system deterioration are ongoing. So, you can see a persistent decrease in CD4 cells, which is the most measurable aspect of this immune system destruction. Plasma viral load, on the other hand, will appear to have stabilized at a particular level or set point. So, this set point is established correlates directly with the progression to AIDS and its morbidity. Here is the surveillance case definition for HIV infection among adults and adolescents, which we discussed in the previous class, which was 1993. And here is the stages according to the laboratory evidence of based on CD4 cell count. So in stage 1, you can see CD4 cell count of greater than or equal to 500 cells per mm cube. In stage 2, CD4 cells within 200 to 499 cells, and in case of stage 3, CD4 cell count is less than 200 cells, and stage unknown. In that stage, there is no information on the CD4 cell count. So based upon this, we are clinically assuring that the patient is having HIV. And Centers for Disease Control and Prevention 1994 revised the classification system for HIV infection in children younger than 13 years. So here is the classification: immunologic categories and immunological categories based upon the signs and symptoms, mild, moderate, severe, and also you can see the immunological categories based upon the age, that is 12 months, 1 to 5 years, and 6 to 12 years. So for this you can refer the DPRO handbook 9th edition. And this is all for today. The treatment part we will discuss in the next one. Thank you.